sorts of things, but they're all sort of based on the concepts that you've learned up until now. And so this lecture that I'm going to give today is actually one of those sort of intro lectures still. And what I'm going to talk about is really one of the core concepts that have infused how people think about how the brain works since the 50s. And really, if you go further back since like the 1900s, like 1910 or something, and that is this notion of the receptive field. And you see this notion everywhere in neuroscience. And so what I want to do is give you a bit of a historical background and also show you how this idea of the receptive field really works as a tool for us to understand the brain and how it can serve as a sort of model building tool. Yeah. So who of you have seen receptive fields in their biology curriculum before? Anyone? No? Good, because then I'll be saying something new for a change. So you all know, of course, also from the previous lecture, if you have a deep neural network, like a convolutional neural network, right, you have these nodes in there, and people call them neurons, right? That's all nice and good. They connect in this sort of convolutional way with one another from layer to layer. And you've also seen that, or have learned previously even, that if you have a neuron, if you call it that, right, a neuron there, it has an activation which you can relate to image space. Yeah. So you can have an activation in one of these neurons, and it means that something is happening in the image, right? Because it actually relays something happening in the image to the rest of the network, right? To all subsequent layers. Yeah. So that's really already a concept that we take from this from biology, and that is the notion of the receptive field. I have misplaced my, I think, yeah, some TSA person or something took it, but I've misplaced my laser pointer. And so what I'm going to do is sit down at my laptop, be boring, not walk around, and just try and go through the lecture while sitting down. Yeah. So if you have any questions, right, immediately break in. Yeah, this is really, this receptive field concept is really something that we depend a lot on. And so what I want to do is first show you how it works, and then go quickly into how we use it in human neuroscience experiments. Yeah, because we're interested in, the course is called Neural Models of Cognitive Processes. And so what's important for us is to try to use this receptive field analogy as a model for cognitive processes. So let's see. Yeah, so one thing that's central to this notion of the receptive field is that of information encoding. And what that means is, because it's just a word, right? But what we mean by that is that if we have a recording of brain, right, and that can be in terms of some sensor that we have, like a MEG sensor, for example, or it can be a neuron, like if we do single cell electrophysiology, we're poking into a brain, like we saw with the parietal lobe response neurons, right, the decision-making neurons. So that's a neuron that we're then looking at. Or if we're doing fMRI, we can record from a voxel, right? So that's a specific location in the brain that we use fMRI to measure the blood flow in, right? And we think then that the blood flow relates to the neural activation inside that voxel, so at that location in the brain. And so what we want to do is have this recording, right, this measurement, and then be able to model what type of information processing is happening in that neuron or in that population of neurons that we are sampling with our measurement technique. Yeah? And specifically, we want to have a model of information encoding in the way that that population reflects. Yeah. So in the literature, what you often see is people use this term. We have an encoding model, which is we try to relate responses that we measure to some information processing characteristic, how information is encoded. And then the idea is that subsequent brain region, for example, that has access to this information encoding stage will be able to take many different cells inputs together and then be able to decode what a population what its computations, right? And also for us as scientists, if we can measure a whole population of electrodes, of voxels, of neurons, what we're interested in is what that population does in terms of how it encodes information in some space as a whole. And what that means is we want to decode from that population. Now, the idea here is that if we have a very nice encoding model for every unit that we record, we can put all of that together, and we as scientists can then use that information to decode. And I'll give you one or two examples of how we can do that. So here, the concept, the main sort of concept really is, if we understand what makes each unit that we record from tick, right? If we can formulate a model that explains the activations that happen there, then we can use a pattern of activations and decipher what a brain region is doing. Yeah? So this is a model of encoding, which gives us a means of decoding. And this type of decoding is different from the types of things that you might see in the literature sometimes, where people say, yeah, we train a decoder to distinguish patterns of brain activation, and the decoder will tell us whether something is moving to the left or to the right, or whether someone is going to choose an apple over an orange. But those are usually very sort of data-driven types of models. There's not really an explicit encoding model oftentimes in that type of thing. And so you get like 55% correct prediction of whether people are choosing an apple or an orange. That's really not the type of thing that I'm talking about when I'm talking about decoding. Here, it's about using an explicit encoding model to perform that decoding operation. And you can do much more beautiful things that I'll go into in a bit too. Yeah? And so, in this field, and I'm going to use here the visual system as a sort of exemplar system, but in this field, the concepts were all sort of charted. They were pioneered in vision science. So what I'm showing you on the left here is a very famous diagram of how the visual system is connected. It was done by tracer studies in monkeys. So that's a horrific business where you inject monkeys with tracers, usually viruses that then go through the neural connections, and then you have to like see how they end up. They did that a lot. This is from the Velleman and Van Essen study, where they did this, and they put together a lot of different sources of information. And they were able to say, like, okay, at the bottom here, we have, where are we? So yeah, so here at the bottom, for example, here we have the retina, and then we have thalamus, and then we have V1, that's the first visual region of the brain. And so you have this entire hierarchy of the visual system, like so, like more and more complicated processing happens as you go up to the top. And here in this sort of diagram, usually the hippocampus is at the top. And I'll get to the hippocampus and the way that it represents information. It's sort of receptive fields, if you will, in the next lecture. But this is a nice way of going there. We're going to start low here, and we're going to trace receptive fields as we go up the visual hierarchy that you see on the left. And what we know happens along this visual hierarchy is that the visual processing starts really, really simple. Yeah, what your retina does is it just picks up, blotches of light, spots of light and dark, and that sort of thing. And as you move up this visual hierarchy, what you get is that information becomes less local. So it becomes invariant to specific locations. And I have a whole list here, right? So what I say there, increased invariance is invariance to where a specific stimulus is. A specific neuron that likes faces is so high up this visual hierarchy that it will respond to a face regardless of where it specifically is in the visual field. And if you look at the there's actually a sequence of face processing regions in this hierarchy. If you look at the bottom, the cells really, really care about what the orientation of the face is, whether the face is, pointing towards you or just a bit away from you. Whereas at the top, the cells don't care anymore. So they become more and more invariant, and they become very specifically activated for a specific function. So they become increasingly specialized. That's what that refers to. Yeah. They also become more and more sort of capable of taking together different types of information. For example, these very high-level facial regions, they put together auditory information also, because when you're looking at my face, you also hear my voice, and they go together, right? And it makes a lot of sense to fuse these different sensations together. And the higher up you go in this type of hierarchy of processing, the more this type of information processing from different senses is blended together. So multisensory integration also increases. And as you go up this hierarchy, temporal integration also increases in duration. And what I mean by that is that in the retina, you show a very quick flash of light, and the response in the retina is immediately gone. If you go to V1, the response to that same stimulus will linger for a bit longer. Yeah, so it actually integrates over a longer time period in V1. So if you go up that hierarchy, and as you go up this hierarchy more and more, this window of temporal integration becomes longer and longer, because at the timescales that the hippocampus, a memory-related region, is interested in, it needs to take together huge swaths of time in order to perform its operations. Yeah. And so you see that there's a sort of increased abstraction in general going from the bottom to the top of this sort of hierarchy. Yeah. And so what I'm going to do, and so really the goal for this lecture is for us to start low and start looking at these very, very basic types of processing that happen in areas like V1, the first visual region of the visual brain, and then move up and see how this idea of a receptive field can be instrumental in our understanding even more complex types of cognition. Yeah. So really, this is like a modeling tool that we can wield. This analogy of the receptive field. And so I don't know what just happened. There was a transition. Anyway, so in the 50s, I think this was at Harvard, a bunch of neurophysiologists were interested in how the visual system worked. And they were working with anesthetized cats. And what they did is they would put an electrode into the first visual region of the cat, and then see whether they could find single neurons, right? So when you are looking for a single neuron, you play what happens in the electrode, you play it on a speaker, and whenever a cell fires, you hear a click. Because the cell firing is an action potential, and it's a very fast changing signal. So it sounds like a click if you just play it on the speakers. It's just an electrical signal that you can just amplify and put on a speaker. And so they had no idea really what they were looking for necessarily. They knew that this was a visual region, but that was about it. And they were trying to show this cat images. So they used a slideshow projector. So they would have this, play images there. And they noticed that a lot of the cells, they just fired only when this sort of slide projector would move a slide out of the screen and a new one into the screen. So whenever there was a strong transient of black and white, and then they were like, okay, we need to find out what causes this, right? What is really the thing that makes these neurons tick? And so what I'm going to show you now is a movie of their experiment that they started doing based on this observation that they had, which earned them a Nobel Prize in the 70s. And it's really compelling. I'll keep talking over the sound. So the first type of cell that they found were simple cortical cells. And you hear these clicks already, right? So they're recording from a single cell. And you see that what they did is they changed their slide projector to something that they could actually orient and move around, like this bar thing. And what they found is that for this cell, it's very clear, right? When you show that white bar at a specific orientation and location, you start hearing this cell fire. And They also show that if you have a region of space just beside it and you show the stimulus there, you don't get any response. See, they're just right beside it. Yeah? It makes really like no sense. Right? That one, like a few millimeters apart, you have this stimulus that makes it fire maximally, and a region where it fires almost not at all. Yeah? And so here they delineate that also as a sort of negative region. And you immediately hear when the bar passes through that positive region, you again hear these action potentials. Now, what this means is that this cell lights a very specific constellation of things happening in the outside world. Right? So only a very specific pattern of visual inputs landing on the retina will cause this cell to fire. And in this case, if you see it, this orientation, it doesn't care at all. It cares a bit about this orientation, sometimes a bit, but if you put it in the right orientation, it's going to fire like a maniac. So this very, very specific pattern, it's a very specific location on the screen and on the retina, and it's a very specific pattern of input that this cell likes. So that combination is what we call the receptive field of this neuron. So the concept here is that you have very strong firing related to a very specific part of the visual field, a very specific constellation of features. And this cell as a whole then is thought to constitute, when it fires, it reports to the rest of the brain that this specific pattern is on the screen. What this means, though, is a couple of things that I want to highlight here. One thing is just experiential, right? You just experience stuff, right? You're looking at this lecture, you're looking at this movie, you're listening to sounds, right? All of that is being conveyed by millions and millions of cells doing exactly this, right? So you're now listening to a single cell, but your experience, the thing that you're experiencing right now, is this orchestra of cells clicking away like this, right? That's what you're experiencing. This electrical activity, this sort of really weird flurries of activity is what you're experiencing. And so these neurons are connected to each other in this sort of convolutional neural network type way. And that's where we, there you see it doesn't activate when you walk out that positive part. So this is how neurons communicate with one another, and we can understand what each single neuron is doing if we understand how it relates to the outside world. We can trace its pattern in the outside world. Yeah? Yeah, these are horrible experiments. The cat is it's very these are like very 50s experiments, right? When they were people were still allowed to torture humans and I don't know what, the weirdest stuff happened. In this type of experiment, to be clear, this is called a terminal experiment. So a cat is anesthetized and never wakes up. And so they perform a craniotomy and they straighten they put artificial lenses in front of the eyes. They fixate the eyes like mechanically with suction cups so that they know exactly where the cat is looking. And then this type of experiment takes 2.5 to 3 days, where they have like teams of physiologists trying different locations in the visual cortex. And at some point, everybody's really tired and they sacrifice the animal is what it's called. And so for what they do is they, for every electrode that's gone in, they used to, and nowadays they have different methods, but they would like burn a trace. So they would actually crank up the voltage and actually burn the trace where the cell, where the electrode was, so that they can perform histology afterwards. They can know exactly where they were recording with their, where every cell was in the brain. But so this is quite horrible in terms of, yeah, so nowadays, a lot of the, this is a very good point. We're now interested in different things also, right? So we tend to think now that we're more interested in more high-level things. And this is very basic, like how does a single neuron transduce visual information? We think we know most of that, yeah? And so people don't do this type of experiment that much anymore. And nowadays people are much more interested in like high-level cognition type things. And we know that we can do those things with monkeys. And so we now do a lot of experiments where you train a monkey for a really long time, right? And so you know, you're much more invested in the quality of life of the animals that you're working with, for example. And they yeah, there's actually like animal ethics committees nowadays. They demand that they require that animals have weekends, for example. So you can train a monkey for in a given task, and then you can do your recordings, right? And usually it's set up such that you can do so much more chronically than in this type of case. Yeah, then you can record from Monday to Friday, and then afterwards the monkey gets a weekend. Right, so that sort of thing has changed very dramatically. At the same time, people have moved away from monkeys more towards like mice. So rodents is now really like a big deal. Turns out that rodents are much smarter than we thought. You can teach them pretty complicated tasks, and yeah, they're much more easy to work with. So yeah, that has changed. Was that an answer to your question at all? Yeah. No, so so the conference that I was just at is like 50% rodent nowadays. It used to not be, but it's yeah, a lot of things that I'm showing you here. People would do more of that in mice now. Yeah. Yeah. Oh, I could keep talking about this, but I don't have slides for this sort of thing. Maybe towards the end of the course. Yeah.
That they were monitoring one cell, but when the animal is is given anesthesia, does that not have an impact with what it sees? And
very good question. Yes. So yes, much of the brain becomes much more silent in terms of this neural firing than under anesthesia. So in many regions, especially the ones higher up that hierarchy that I was talking about, you cannot record from if you have an anesthetized animal. So they are completely silent. You can show a stimulus, but you would get maybe 1/10 of the response that you would have if the animal were awake. So definitely, the point here is that V1 is so low, right? It's so close to the retina that even though the animal is anesthetized and is like literally unconscious, right? These regions will still be stimulated in more or less the same way as they would in a behaving animal. And this has all sorts of implications for what you think being conscious of something means, right? Which is a whole separate field that I'm afraid I won't be able to go into. But that's a really good question. If you anesthetize, a lot of the brain starts functioning completely differently. But these regions, being so low in this hierarchy, are much more mechanical in the way that they process visual information. So they're not influenced this much by things like the consciousness, if you will, of the cat in this case. Yeah. Yeah. So just keep asking these questions, because I think it's good if we frame. You get a different viewpoint for every question. Now we call this is vision, right? And because there's a couple of reasons why we focus on vision in many cases in neurosciences. The first is that we are very visual monkeys. Yeah. A lot of our brain is devoted to vision, and in terms of how most of us experience the world, vision really is a very dominant modality of information. We imagine things. That's because we have a mental image, right? And that already says something about how our inner lives reflect this dominance of vision. So that's one reason why vision. The other is a lot of the brain. So as a consequence, a lot of the brain is devoted to vision, like half of it or something. And that also means that people have started finding out stuff sooner, right? It's a reason why there is much more of a tradition in terms of science for vision than there is for example, our sense of smell. If you want to look up literature for our sense of smell, there's like maybe 100 papers per year. if you look at like vision, there's like thousands of papers per year. Right? So there's much more of a body of literature there. So vision is very dominant. And so we know a lot about vision. So what I'm going to show you now is all vision. But I'm going to move to other modalities too during the lecture. Yeah. So if you look at these visual receptive fields, we just looked at a simple cell, as they call it. I'm not going to go into the whole sort of family tree of different types of receptive fields that we know, but here's just a few examples. So if you go in the thalamus, which is a region one level down from V1, right? So signals have to go through LGN to hit V1 to go to V1, and there the receptive fields aren't these beautiful oriented patches, but they are these sort of circular patches. So there's like a center where this cell fires, and then there's sort of a surround where it inactivates, yeah, similar to those the pluses and the triangles that they drew, but then circular. And only when you go to V1 do you get these oriented receptive fields, where what we see here is that at the level of V1, what the brain is starting to do is assess the orientation in images, right? The local orientations in images, which of course you need to be able to, for example, trace a line and see a silhouette. You need to know the orientation at local locations. Yeah. So that's in V1. In V1, also over time, you get receptive fields that like a very specific flickering frequency. So so that is a transition has to happen at a specific rate over time, right? So here, space is this is space, right? This is X by Y. So that's space in the image pixels. Here, it's X by time. Okay? It's just another dimension in which the visual system can encode information, right? And here you see that we also have cells in V1 and a subsequent region called MT, which is very motion selective. The M doesn't stand for motion, it stands for middle, because it's the middle temporal region. But what you see here is that this cell has, again, X and time, right? So it's space and time, space-time. But it has not a sort of symmetric thing over the time axis, but it has an oriented thing over the time axis. And what that means is it likes a specific direction of movement. Right? So this cell, when this cell fires, the rest of the brain that's connected to it knows, at this location in the visual field, something is moving with this speed in this direction. Yeah? And that, of course, you can build up a lot of different types of information from, right? If you're driving your car around traffic, this is really the type of information that you're using. Right? Oh, but then you have like millions and millions of neurons reporting similar things and build it up from lower to higher levels. Okay, so we have all these visual receptive fields, and we see the types of processing that change as you go up that visual hierarchy a bit. And you can take this further, like we can see this huge neural network of different types of receptive fields. In this case, it's about space. And we see this network reflected in the human visual brain. This is just a precursor to something I'll explain in a bit. But we have this entire sort of layered network in our brains, starts out with low-level processing, goes towards higher-level processing. And this type of hierarchical system really was the basis for this design of these convolutional neural networks. Yeah? So really, the inspiration for these deep neural networks that we use for image recognition came from this architecture of the brain. Now you might think, okay, well, what is all this like colors and what have you on this surface of a brain? I'll show you how that works, because one of the ways in which we can use this receptive field analogy that I've been talking about, right? So what I've shown you up until now is all very concrete receptive fields. Yeah, just basic, like what is a receptive field of a neuron? What I'm going to talk about now is how we can take that idea of the receptive field, of finding something in the world that makes the unit of measurement tick, right? That causes an activation. How can we take that to a human experiment? Okay? That's the first sort of abstraction from this receptive field idea, and how we're starting to use it as an analogy. So what we can do is we can map also in humans. So we can do this in animals, but the nice thing is here we can do fMRI in humans. And what we can do is we can map out these retinotopic maps of the human brain. So every level of that network that I'm talking about actually mirrors the image that you're looking at. It mirrors visual space. And that visual space is encoded in retinal coordinates, of course, because that's how the things come in. We call that retinotopic. Now, the idea is that we can put someone in the scanner and then perform a very, very simple experiment that allows us to map out all of these different layers of that convolutional neural network that we have in our brains. And I'll show you two different images here, two different movies, one on the left and one on the right. The left is what happens on the screen, and the right is what happens in the brain. Yeah. So I want to invite you to just look at the structured visual input first. Right, so there you see this bar, very flickery. It moves across the screen. If you then look at the what happens in the brain, oh, it stopped. Jeez. So you see that there's actually a wave of activation going across the brain. So again, this might be a bit vague. So what we're looking at on the right here is a brain that was. So you have a brain. It's in 3D, right? Then what we do is we can take the gray matter surface of that brain, we can inflate it like a balloon in the computer, of course, because we don't want to explode people's heads. And then we can cut it up and like flatten it out like so. Yeah. And so what you're looking at here is the left hemisphere on the left and the right hemisphere on the right. And the visual cortex is here in the bottom. The parietal cortex is here and the frontal cortex is here on the outsides. Yeah. Now, the most important thing to see here is that when I show something like a bar on the screen and it moves like this, what I also see, geez, oh no, I have to quit this now and restart it because I was going to show you the actual experiment, but it's horrific. Yeah, so what you see here on the right is that really this wave of activation in red that travels across all of these different maps in the visual system, right? So when you have a wave that you're causing on the screen, you also track these waves in the brain. And then the question becomes, can we model these waves in the brain to understand how the brain represents visual space, right? Can we, for every location on the surface of the brain here, can we model what it's doing, like a neural model based on a receptive field analogy of the cognitive process of vision, if you will? Yeah.
The first look speaks and the point doesn't track the line.
Exactly. Yeah. So that's really important because whenever you move your eyes, you're going to move the image on your retina, right? And if this processing is fixed to your retina, yeah, then you're jumbling things up, right? And you don't want that. Yes. So people have to be fixating the center of the screen in this type of experiment, which is also very limiting, right? Because in many types of natural behavior, we do move around, not just our eyes, but also ourselves, in which case you can't do that in a scanner anyway. Okay. So this is a sort of experiment that we then do, right? So people fixate in the center, right? And in this case, people performed a task on this bar because we were interested in what attention does, and maybe I'll talk more about attention in a later lecture, like how can we model attention, that is. But here the task was, you can always see it in this bar. Sometimes it's more yellow and blue, and sometimes it's more red and green, right? And so that was people's tasks. They would be required to say, is it red or green or blue and yellow, right? And you can see, there you have it. You can do this, right?
You need to do that when you are fixating.
Yeah, while you're fixating, yeah. And it's harder when, yeah, that makes it quite a lot harder because you want to look at it, right? Yeah, one of our PhD students.
Confirmation, I think, because you do not want to be wrong.
Yeah.
And you want to make sure that you are pressing the right.
Mm-hmm. Yeah, so you move your eyes just to check, right? Yeah. I do that a lot too. We're now, yeah.
Is there a specific reason why it's like why they choose like this kind of? Because it's like noisy and it's like a specific pattern. I don't know. It seems kind of a specific.
It is very specific. So yes. Well, that's a very good question, actually. You know, you see each of these elements, there are actually 2,000 of them, but each of these elements is like a stripy pattern, right? You see that. Well, this is called a Gabor function. It's actually the multiplication of a Gaussian function that I'll explain in a bit, and a sine wave. And this is really the standard model to model these receptive fields. So a pattern like this can have a location, right? That's when you can move it around, and it can have an orientation, and it can have a frequency. It can be like wide or it can be narrow. And so the function that created these parts of the stimuli are actually exactly the same that you use to model these single units. And so what we think is we can use it as a very strong stimulus because of this, right? So each of these elements is actually very specifically causing activation in a specific set of neurons. That's why we used it. Yeah, there you have it. Yeah, it's pretty too, isn't it? It looks like a migraine attack.
It does lead to more activation if you compare it to just a straight monocolor or single color line. Yeah,
yeah. So this is, and it's also moving, right? So all of those movement detectors are also stimulated. Yeah. So, but there's an open discussion as to what is the best stimulus for an experiment like this. Some people think you should just use checkerboards, right? You know, the chess game board like white and black. If you flicker those around, that's also a very strong stimulus. It's strong in a different way than this one. It's very hard to say which is better because it also depends on which region of the brain you're interested in, for example. But yeah, so that was a very good question. It's a nice example too of how we understand how the cells, how the neurons work by means of some model, right? And then we can actually use the model to create a stimulus, right? It's a nice turnaround. So quickly before we have a break, I guess, this model that we're using to understand these local responses in the brain is a very simple model. It's called a Gaussian, which you all may know from statistics. It's the normal distribution, right? So it's the most standard distribution of statistics. Whenever you have infinitely many sources of variation, the result will become a normal distribution. It's very simple. It has a couple of parameters. And let me just show you the entire slide. So this is our model. So in this case, our bar and our voxels, we are not interested in orientation just yet. So we're not interested in orientation. We're only interested in what portion of visual space makes the single location on the cortex fire. Yeah, what causes its signals to rise. Yeah. So what we did is so if you have a model like this, what you can do is you can change its position, right? So that gives you a model that predicts different things. Here, for example, you could place this peak here, and you would change just this X and Y parameter of the model. So those are the location parameters of the model. And you can also change the size of this thing. Yeah, makes it a wider distribution, and that's another parameter. So here you have just the equation for this model. I'm not going to quiz you on it because it's not very important, but this is only part of the normal distribution equation. If you're interested in the response at some pixel, which is defined by X and Y, and your X0 and Y0 give you the location of this receptive field. So you can actually change it around. So the way that we had like a drift rate in the drift diffusion model, we now have position X, position Y, and standard deviation, so size. Yeah? And we can vary these to take this movie of what happened on the screen and produce predictions of what any brain region would do. So the thinking is as follows. You say, I have a model and it really likes things to be on the left top and it's this big. So that's your model. You have 3 parameters that specify this. And then you could say, okay, but if I have this experiment that went through this receptive field in this sequence, I predict this time course of response. Yeah. So this is sort of the sequence that you have. You have a spatial model. You have your experiment here at the left top, and this allows you to produce a prediction. And when you look at what we measure in the brain at a specific location, you actually see that when the bar passes through the screen 8 times, you get this beautiful activation 8 times. Yeah. And this is in a human brain, so we can actually have very, very good recordings of this type of modelable response in the human brain. And what we can do is we can use these fitting routines that you've now learned about, right? What we do is we do a grid search on these parameters, and then we do an iterative search on these parameters using gradient descent, right? So that's just the analysis that we do, fitting this model to the data. And what we find is that we can estimate the best fitting parameters x0, y0, and size that minimize the error that we have in our prediction. This is beautiful, right? So you have this, you have a model of what location this voxel likes in the visual field, and you can explain the responses like really, really well, right? If you look at this, you don't need any statistics further, right? You just know, okay, this is working, sort of thing. Yeah. And the nice thing is, and now we're getting back to those maps that I was showing you just before. If you do this for every location on the surface of the brain, you have this model, you fit it. That's beautiful because, you know, when we talked about, I talked about this Parkinson's study that we did, right? And we were able to see that certain subjects behave differently, and that was reflected in specific parameter estimates, right? And so subjects differed in terms of these parameter estimates from the model. The model really allowed us to say something about differences between patients, and also things about, you know, things like what does the medication do? Well, in this case, we don't have a population of subjects. We have a population of locations on the surface that we're interested in. And these parameters are not explore-exploit trade-off. These parameters become the x and y in size. Now, the x and y, you can actually translate into something we call polar angle. So that's like what hour on the clock face this thing likes. So it doesn't matter what's the distance to the fixation mark, but we only care about this direction. It turns out that's really a nice dimension to look at these maps on the surface of the brain. So we can find like the first visual region, the second visual region, the third visual region, because they all reflect visual space, and we color visual space preference. We can find these maps of visual space back on the surface of the brain. So this is a way in which we fit a model. We get these parameters out of the model fits, and this allows us to learn something about the organization of the brain. And you really see that entire hierarchy of brain regions go up towards even the frontal lobe. Yeah? You really expect things to go like, here you have it like green, blue, red. That's a full map of the image that people are looking at, but in the frontal cortex, all the way in the frontal cortex, we can see this organization of retinal visual space be there, right? So not just in this first region. So this here is V1, and that's exactly that same region that they were recording from in the cat, right? So we have this beautiful way of using this receptive field analogy to also translate these concepts from animal research, single units, to these sorts of what we call population level. So we're recording from an entire population of neurons, measurements in the human brain. And we can then really start to inspect things like how the processing evolves over between these different regions that I won't go into right now. So let's have a break now. Let's be back in 10 minutes. Coffee time, yeah. So this is just one example of, you know, how this receptive field analogy works. And I'm going to take it much, much further. So the second half of the lecture is going to be like, okay, now we have this idea, right? We have an activation somewhere. How do we relate it to what happened in the outside world? And this can be construed as broadly as possible. And I'll show you very broad examples in the second half. Yeah.
Like yellow, though.
Oh yeah. So if you were to look at the other hemisphere of the brain, yeah. So you could flip the color scale, and then you would see. Yeah. So these are completely mirrored maps from left to right. Yeah. So what you see is that
What one of the eyes takes going on.
Well, that's a really good question too. No. So when you if you look at the low levels of the visual system, the left image and the right image, they aren't separated according to eyes, but there's a cross. So in the if you have the optic nerve, right, it doesn't go straight from right eye to the right hemisphere and left eye to the right, to the left hemisphere, what it does is it crosses the nasal portion. So half of the ocular nerve, of the optic nerve, actually switches sides. And what that means is that your right hemisphere, it takes the left visual field from both eyes. And your left hemisphere takes the right visual field from both eyes. And so that's also why this is visual space and not eye. If you're interested in eye, so this does mean that for the left visual field, both of your left hemisphere, your right hemisphere will get all of the information from your left visual field, yeah, but it will get it from both eyes, yeah. And so what that means is that it's able to combine the information coming from the left and the right eye. And what you see is that if you zoom into this map, I actually have this map for this subject also. It's me, so there you have it. Okay, but what you can here in V1 in this first region, you actually have, if you zoom in further at the millimeter scale, you actually have patches: left eye preferring, right eye preferring, left eye preferring, right eye preferring. We call this ocular dominance columns, and we can use fMRI to actually map them in single individuals.
Well, I know there are like those interesting experiments with like just patients that don't have one of the hemispheres.
Oh yeah.
And those that have the right hemisphere can kind of see the both sides. But for the left hemisphere people, they kind of don't even know that's the what is it, their left side.
Yeah, exactly. So that's a good point. So these regions here in parietal lobe, they're very involved in attention. And what you see is that for those regions, the right hemisphere actually can cover the entire visual field. So it sort of takes the right visual field also, whereas the left, it's still very lateralized. And so at this level of the parietal lobe, we have a functional specialization of the two hemispheres. This is also why people say like the right hemisphere is a spatial brain hemisphere sort of thing. That's because of this. If you have damage to right parietal lobe, it's much more debilitating in terms of orienting yourself in space than it is on the left. So left does much more like yeah, in most people more like language related processing, people think. I do have to say that this sort of common sense thing, right? The common knowledge of the two hemispheres being completely different in terms of what they do, that has been, people are thinking that less and less. So the better we become able at understanding the nitty-gritty of how the brain processes information, the more symmetrical it does seem to be. So there are asymmetries, but in many cases, it is also still pretty symmetric.
Well, if there is any like asymmetry, like big symmetry will be like between the hemispheres, but out there. Connected like that, between the muscle is hemisphere. And yeah, basically talks about this kind of how people draw. Like they don't draw like certain things. They don't draw the left side, but it's very detailed.
Yeah, exactly. I like these stroke patients that if you ask them to draw a clock, they'll put like all of the numbers in one half of the clock or something, right? There's one of those standard, yeah. Yeah, sure you can. I'm still getting, I think I need to like stop this. Stop the, I'm also, great. Now, these different parameters, you can sort of do different things to them, right? So for example, we have an X and a Y parameter for these models, right? Because they shift like, in the vertical and horizontal direction on the screen. But what's interesting, of course, is another dimension that you can distill from this. That's the distance from the fixation mark, as we know that's a very important thing. If you think about visual space, right? If you just think about what you're experiencing now, it's very sharp in the middle and it's very, very blurry in like in the outside, right? And so this axis of the distance from where you are looking is very, very important. And we call that eccentricity. Something is more eccentric when it's in the outer parts of the visual field, as we call it. And it turns out that that is really a sort of primary dimension of how the visual system is organized. And it turns out that you can analyze things like the size of these receptive field models along eccentricity, and then you see that these sizes increase as you go up the visual hierarchy, which is also the case for convolutional neural networks, right? They, because of this sort of max pooling type thing that you do, you get really like smaller representations in the upper layers, right? And that means that every single one of them, every single unit at those levels actually has a much larger span on the image space. So this is really something that you see happen a lot in the visual system, right? You see the axes here, right? It goes from 1.3 to 3.5. It's like a threefold increase. So it really provides you with a way to quantifiably understand what the visual brain is organized like. Yeah? And you can take this much further, right? We started the course with these signal detection theory models, right? Very, very simple, a couple of parameters that you distill from the measurements, the way that you also distill parameters from measurements here. And what you can then do is take the simplest possible model and start to make it more complicated. Now, one standard feature of this type of receptive field model is that the simplest possible version is a linear version. And you can actually make them nonlinear by applying some sort of exponentiation type thing. So that, for example, if you show a stimulus of strength 10, you get a response 10. You show a stimulus of strength 100, you don't get a response of 100 because then all the neurons would explode, but it sort of dies down slowly. That sort of thing, we call that a nonlinearity. Similar types of processing are actually implemented in convolutional neural networks also. And so we can take this simple model and make it more and more complicated, right? And here I'm just showing you two examples of how you can complicate things. We're going to talk about a specific computational principle called normalization later on in the course. And that will be one way, that's really an example of one way of taking this simple model and taking one step in an important direction to make it a bit more complicated, but then capture a lot more in terms of responses. Yeah. So main point here is we start with a simple model, and then we can start making it more complex. We can start building more complex models based on a simple model. That's really always the way it works. Now, one thing that you tend to do in this sort of case is that you want to also decode from the model that you're fitting, right? I talked to this, the first slide was, okay, we want to characterize information processing as encoding of information. That's what we've done now with this model. Now we know how the brain encodes information. Now we want to sort of invert that whole thing and understand how sort of decode what the visual brain region is doing. So this is like one of the ways in which we do that. So what you do is you can fit all of these receptive field models on many runs, and then you have a separate run that you want to decode. So that we call a test run. So what we have from that training set is that we have all of these different receptive fields, right? They all have a different location, all have a different size, and they all span together. They span visual space. And now we have a bunch of time courses in a test set. And what we can then do is sort of invert this encoding model and actually decode what a visual brain region is seeing, right? So what is the most likely stimulus that we can decode from a brain region? So this is a beautiful trick. You actually can create these movies of what a brain region thinks is on the screen. And that can be a very compelling sort of reference frame in which to present your results, for example. So this is just an example of how you can take this encoding model approach, this receptive field approach, and then really try to understand things at the level of the representation as a whole, and that we call decoding. Now that's all nice and good, but I'm going to take a step back and just keep talking about this encoding, because this receptive field analogy is really where I want to what I want to focus on most. And then I'm talking about voxels here, but the same would go for like any recording that has multiple that sort of fuses the results of multiple neurons together. And so I want to talk about this one extension of this receptive field model, where these authors actually extended it to also incorporate orientation and this spatial frequency, so the fineness of the pattern. So what this means is, here they're actually doing an fMRI experiment very, very similar in terms of the results to that Hubel and Weasel example that I showed you on the very first slide, that movie, right? So we're adding, not just, so now we don't have just location in the visual field, but we actually have orientation in there too. So it's very similar. And what you then do is what in this case they did is show a bunch of images to subjects. And then you can fit this sort of receptive field structure and start predicting responses. So what's the output in this case? What does a model output look like? These parameters, what does that mean? Well, so this is exactly the same as what we saw before, right? We have like this receptive field location, right? So this is visual space, and this is the location of this specific voxel's receptive field. It's just fit a bit differently. I won't get into the specifics, but here they're also adding orientation and spatial frequency. So you know that if this voxel is highly active, not only is there going to be a salient part of the image in that location, but also it's very likely to have this orientation and this spatial frequency. Yeah? And you can see how you could use that also in sort of a decoding type thing, where you wouldn't be decoding just, you know, where is a bar on the screen, but you'd actually be interested in being able to decode much more complicated image characteristics. But you can also decode a bit more differently. What you can do is try to identify which image is being shown from a whole set of images. So you take the pattern of brain activations and you're going to say, okay, which of these 100 images is being shown at this moment? And if your encoding model is really good, then also this decoding, this image identification should be really good. So what you do is you say, okay, what is the predicted voxel pattern for my model based on this image? So which of these predicted voxel patterns based on my model looks like the actual activation pattern? Yeah, you just rank order them, and what you get is that you can really well predict what the bold pattern will be. So that's the pattern of activation in a given region, and you have a very similar, very simple measure, namely just the correlation between the predicted and the actual time course. And this type of image identification really is very, very good. So here you see that they're choosing the correct image in like 96% of the cases. So here's a few errors. There's one here, one here, one here, one there. Otherwise, they're all on the mark. Yeah? So because you know what every single voxel, every population of neurons in V1, in this case, is doing, you can actually interpret what the pattern of activation in that brain region means, right? Because you know exactly what image created it. This sort of thing, actually, you can extend pretty drastically, right? So you can look at your identification performance if you extrapolate, and it means, in this case, for two different subjects, that you would still be at around, well, let's say 70% or something on average, if you were to show 1,000 images, right? And if you're okay with 10% performance, you see how it starts to become like an AI type problem that you're performing on a brain analysis, right? If you extrapolate to 10% performance, so if you're going to be okay with 10% performance, you would be able to encompass a set of 10 to the 11th of images, right? So you're really starting to actually get a grasp of image space by virtue of what you know this brain region is doing, if that makes any sense to you. Yeah. But you can go even further. I showed you that these motion selective neurons, right, in the visual system, they have like a motion direction preference. Right? And what that means is you could actually also model this, but then you just need to show people movies. Yeah? So if you show people movies and you extend this model to also have movement selectivity in it, you have an encoding model and a computational model of how every location in the brain computes to specific types of visual information, in this case also motion, and you can do this reconstruction based on it. And this is already very old, But this is an example of what you can do then, right? So this is similar to that bar that I was showing back and forth, but then based on actual images, and then reconstructed from brain activation. So this is actually already, I think this one is 12 years old. So I'm pretty sure that nowadays we could do a lot better. And there are examples actually where we can do a lot better. There are people that have done this type of experiment with faces. So they're not interested in movies in general, but just in how to reconstruct faces from brain activations. And then they can be like really good. Like you can't even tell the difference between what you're able to reconstruct and the actual image that people are shown.
The faces are the most well reconstructed.
Hmm?
Even here, the faces are the ones that are most.
Yeah, yeah. Yeah, definitely. You can definitely already see that there's a face here, right? Yeah, yeah, I agree.
We use like existing pictures and video files to reconstruct the videos and
Yes, we can, if you want, we can get into that at some point. But yes, so here, if you do this in a Bayesian way, for those of you that are interested in this sort of thing, I'm not making this whole thing part of the course, but we have a whole, we're developing these types of tools. So what you want is to get the sort of a posterior distribution in image space given some activation pattern in the brain. Right, and so it's not just a single movie, but it's actually a whole distribution of possible movies in sort of what you could call movie space. And what they've done here is they've taken YouTube and then sampled like this movie, this 1 second clip. What is the likelihood of this image? Or the probability of this sort of movie clip, given the bold pattern, given the brain activation pattern. And if you do that for 150,000 movies, each of them has a probability attached to it. And what you can then do is average the movies together, weighted by their probability. And that's what you're seeing here. Right, so they're sort of using YouTube to sample the posterior of possible movies, given this brain activity pattern. Yeah. This is like, I don't know. We can get really technical, but that's not really the goal of the course. But that's, if that made sense to you, then we can talk more about this later also. Yeah.
So that reconstructed video was not based on the present clip, but based on many clips.
Yes, and so many clips in two ways. It was created based on many clips, as in this averaging procedure that I just outlined, but also there's a model in between, right? So this actually was never seen by the model, right? So you just have a pattern of brain activations. To estimate this model, right, to understand what this pattern of brain activations even means, you also need a lot of movies, but those are just for training the model, for being able to fit the parameters of the model. Yeah.
Just one last question. The correlation matrix that you showed, it was based on just the orientations of the images, right?
Yeah, local orientations. So
it's like straight lines, or don't have to be straight lines.
Yeah, so local, right? So certain every sort of voxel, every patch of cortex that you're sampling from, they will have some receptive field somewhere in the image, right? And they won't just have a location, but they would also have an orientation, right? So you'd be able to distill whether, you know, if you're looking at a big picture of the sun, right, you would expect this circle of responses, but only for those locations that actually have an orientation preference that outlines according to the circle, that is tangential to the surface, to the circle. Yeah, so both location and orientation feature into that. Yeah.
It's just like little lines.
Yeah, exactly. Yeah. Yeah, I think I have an example of that in the second tutorial, the second practical, which goes into like what is the transformation that you get, right? So what V1 does, because it has this orientation preferences for every, yeah, and it does
and the thing that we focus, it's in the back. Yeah,
exactly, in the back of the brain, you mean? Yeah. But Yeah, so specific, you can orientation filter an image, for example, and in essence, that's what the brain is doing all the time. So you can actually do this also for sound, because we have a tonotopic map, like we do a retinotopic map for visual space. We have a frequency space in auditory cortex. And it's not just frequency that it likes. So just to be clear, this goes from black to purple to green to light green. So that's this whole thing here. But it also goes the same direction like here. It looks a bit patchy, but it's I can assure you there's more beautiful maps somewhere. And so what you can then do is do the same trick, right? But now you don't have a movie, you don't have a picture, but now you have a sound, right? So I have these sounds here that I'll try to play. I'm trying to play them. Maybe I'm trying to play them to the screen and it's not doing anything. Wait, like a good teacher would have tested this, right? Yeah, no, it's supposed to be playing this to the an actual sound. That was the decoded sound, right? So you see, for this type of natural sound, it works really well. But if you then try to do this, and this is a nice example, I think, the next one, it's going to be spoken words. So you have spoken words and then a reconstruction of the spoken words, but just based on the primary auditory features. And so I'll just play it.
Beyond all this.
And so what you see here is that in order to replicate or reproduce the sound of a human uttering, you need much more information than just the basic low-level sound characteristics, right? There's a lot of lingual stuff in there, a lot of information, like implicit information about pronounceability, you know, a lot of language information that already also goes into that, right? So it tells us something interesting also about what the brain must be doing to analyze speech. It's not just low-level sound. Okay? In any case, I'm gonna try to speed up a bit because I think, yeah, well, I only have finite time. What you've now seen is that we started out with just visual space. We then added orientation, yeah, and we already knew that we would be able to add orientation. Then we added motion, yeah? And now we went to sound. It's all the same. There's a receptive field. Yeah? Every location in the brain we can estimate a receptive field for, yeah? And then we can decode. And decoding is just a way of showing you how the encoding worked and what types of features it was sensitive to, like in the speech example. Yeah. I want to again take a step back away from the decoding and towards the encoding part of things, because this analogy of finding that in the outside world which causes activation in this recording is, you know, finding the receptive field of the recording that I have, be it a single unit neuron, like an MEG sensor, or a voxel in fMRI, that doesn't matter. We can try to now find the space in which this receptive field lives. Yeah, so we're going to divorce this receptive field notion from visual space, from auditory temporal frequency, or like a frequency of sound. And we're going to now see, does this sort of thing actually apply to arbitrary spaces? Like, can we find spaces in the brain that we have apparently receptive fields for? And so this is a beautiful example of that. It's one of our collaborators at the Spinoza Center. He found this. There's a very, based on a single finding in monkeys, where someone had shown that there are specific neurons that like the number 1 better than the number 2. And then there are also in the same region, there are neurons that fire more when there is the number 3, then the number 2, then the number 4. So they seem to be tuned to a number. And so they set out to do an fMRI experiment in humans to see whether this is the case. Can we find receptive fields to numerosity in the human brain, right? So this is done by fMRI, but the receptive field analogy holds. So that's the backdrop And what you see here is that they did an experiment where they varied numerosity, right? So from 1 to 4 to 7, they actually went 1, 2, 3, 4, 5, 7, and then 20, and then 7, 6, and so forth, and so forth. And they did that by keeping all sorts of things equal. So this is then constant area, this is constant dot size, this is constant circumference, this is density changes, this is variable features. They just wanted to know that they were looking at numerosity and not something that correlates with it accidentally, right? Not a confounding thing. And then what they saw is that if they look at specific locations in the parietal lobe, in this case, you see right next to each other, so really right next to, like a half a centimeter distance between the one voxel response and the other response, you see either a temporal pattern like this, or a temporal pattern like this. So this one is very active right at the start of this experimental run and right at the end. And this one has this weird sort of bimodal thing peaking near the center. Now, the only way that you can explain this sort of really weird profile of activation is if you assume that the neurons in this voxel that you're recording from have a very specific preference for some numerosity. And you can actually create a receptive field model to describe this sort of preference, where for this one, it peaks around 1, and it has a specific width. So that's when this one and this one happened, it has the highest activation. Yeah? What you see here is equally a set of neurons that you sample with a voxel that have a specific preference, but there the preference is very high and it has a very, very broad selectivity range, right? And so you're able to capture this, this pattern in the data that's pretty intricate with this type of receptive field model. So this analogy seems to hold at least for these two voxels, yeah, for these two recordings that we had. But if we then look at the whole brain, or at least like patches, like we saw these maps, the retinotopic maps, right? It turns out that you get these beautiful maps of numerosity preference in the parietal lobe in this case. So you actually go from low numbers preference to higher number preference. Yeah? So and here you have another example in a different subject, also in the same location in the parietal lobe, right? See, it's almost the same location across two subjects, and it goes from low numbers to higher numbers also. Yeah. And so this is pretty funky. This is the first instance that we saw that an abstract quantity, like an abstract dimension, which numerosity is actually represented in a sort of in a map-like way, in a receptive field-like way, in the human brain. And so what that means is that this is a validation of that concept of the receptive field. Yeah, it allows us to you can apply this concept of the receptive field in many different dimensions. In this case, an abstract dimension, and then show that the brain is actually organized according to this dimension. So it really allows us to learn something about the brain's organization. And so one other thing that you can so this is just a way of tracing that map along the surface, right? So this is distance on the cortex going from here to here, and then you see that there's a nice map-like progression, a monotonic progression. That's already what you see in the colors. This other thing that you see is very similar to the thing that I showed you in visual space. You saw these slopes, right? If something is very close to the center of fixation, the receptive fields are small. It turns out that for numerosity, a very similar sort of sampling thing happens. I don't really have time to go into this, but it's called the Weber-Fechner law. If you want to talk more about it, I'm very open to it, but not part of the lecture. Okay, so that's cool. We now have not just a specific model, but we have a sort of analogy, right? We have a way of thinking about what causes brain activation, as in what happens in the outside world that we can relate to brain activations through this receptive field idea. And we can create models that quantify this, and we can learn from the parameters that come out of the models. Yeah, you've learned how to fit these models now. What can we do with those? So I want to get into that for a bit. I still have 15 minutes, right? Great. Because the coolest stuff is still, yeah. Did you have a question also, or did...
Yeah, I was going to ask, but maybe not an important one.
I'll be the judge of that.
Okay. So every time when you were talking about the stimulus, it always comes in a white background or a gray background. Is there also a specific reason for that? Even with the numerosity example that you showed,
Yeah.
you have different structure of the stimuli. For example, some were circles, some of the stars and squares and rectangles or whatever.
Yeah.
But is there a specific reason why we have like not really a catchy color? For example, not bright colors, but these are.
Oh yeah. Yeah. So in this case, so I'm sort of paraphrasing what I think my collaborators were thinking when they conducted this experiment. So just to get back to the specifics of the experiment, right? It's a boring experiment. People need to look at the center of the screen. And the task in this case was not to do anything with the numerosity on the screen, but once in a while, so this would change all the time, right? So it would just be dots would be replaced on the screen every 800 milliseconds or so, right? So it'd just be very boring. You're just looking at the dots jumping across. And then after 5 or 4 seconds, there's 2 dots and they jump across the screen like so. And then there's 3 dots and they jump across the screen very slowly, very, sort of experiment. And your task as an observer, as a participant, is sometimes, all of the times, these things are black. And then once every 10 or so, randomly, they make the dots white. And you have to respond. Now they're white, so that you don't fall asleep. That's the only, you have to look at the center of the screen. Wait. That's the, for like 1.5 hours at a time. So yeah, they were interested in, does this abstract dimension, is it represented in a very sort of low level type way? So these people, my collaborators, they are very sort of sensory neuroscience, right? They're from the visual domain. They don't care about, high level cognition much. This is just numerosity, which they consider also sensory.
But even from the sensory level itself, then how do you say, for example, if the background is like green and then you still can see white and black on that, but why is it always then a white background or a gray background? For this, yeah, I do get it, because you want to between black and
gray
white
is between black and white. That's yeah, and we do the same in vision science experiments, usually like a gray background.
Yeah, because I participated in one of the experiments and it was also the same, but then it was like multicolor and then yeah.
So usually you want to keep things as simple as possible, right? Anything that could have any effect, you don't want,
Okay.
right? So you want to, in this type of experiment, you want to ask a very specific question, and anything that could be in any way possible a distraction from the main goal, you want to completely extinguish. That's just a matter of, sort of robust experimental design. So that's why people always maintain, okay, for example, the grayness of the screen, it's not just the fact that it's right in the middle of white and black, it's also the fact that, it gives you a solid baseline over time. It doesn't change over time. It has no color, so you're not really addressing any color processing that might get in the way. There's all sorts of things that you don't want to happen if you're just focused at that specific question. Yeah. Does that answer?
Yeah,
no.
yeah.
Cool. So I said this group is very sensory neuroscience type group, but numerosity is an abstract modality, right? It's an abstract dimension. So they have a point in there. It's a science paper, so it's it's very nice. But this analogy of the receptive field, it doesn't stop, right? Just a matter of what you put in that receptive field, right? Can I think of a dimension? It could be, how long do I have to wait to get my next Snicker bar? It could be anything, right? There's no pain. Yes, you could have a pain receptive field. Like it could be like, is it an itchy pain or is it a non-itchy? Like you could have like a dimension in pain. Definitely.
But it's hard to measure pain in fMRI, right? Because people will move.
Yeah, exactly. Yeah. So you get into the experimental design sort of issue type thing. But yeah, so I really want to give you this idea, right? We have now this analogy. We have a measurement and we have an outside world that we want to relate it to. How far can we go, right? And I want to argue that there really isn't much of a limitation there, as long as you can, formulate a model and create an experiment that allows you to fit this model. Yeah. So what I want to do is really show you a bunch of examples where people have done just that. And so one of the examples here, I think, is beautiful. It's semantic space. Instead of visual space or auditory space, we use semantic space. And so this is again from the same lab as the movies that they reconstructed. So this is like the follow-up of that movie experiment, where they did more or less the same experiment. They showed people movies for 2 hours in a row. And what they now did was not say, oh, now I have this movie, and then on the right bottom, something is moving to the left, sort of thing, right? Which the visual model with the motion selectivity would do. Here, what they did is they just sat down, watched the movies like 10 times or very slowly, and would just say, oh, now there's this in the image, now there's this in the image, like words, right? So they would denote which concepts were present in the movie at every second of the movies. Yeah? And so what you can then do is, measure brain activity, of course. And you can perform regression from this design matrix, what happened when in the experiment, and just regress it onto the brain. And what you get is these model weights, which we call beta weights. And if you're going to do the fMRI neuroimaging course, then you'll know exactly what that means. But what we get is, for every voxel in the brain, how much does it like the word woman, or a woman appearing in the movie? How much does it like the word, like whenever a car occurs in the movie? And so now what we're actually doing is we don't have a receptive field in the in visual space, but we now have a receptive field in a semantic space, right? And then the question is just, okay, if we fit this model, can we, predict what the bold response will be doing out of set? Because then we're explaining what these voxels are doing. And so and you really see this nice Gaussian shape here also, right? That's exactly what I mean to convey. We're looking at a receptive field, but now the receptive field doesn't live in a sensory space anymore. It is now abstracted, and it lives in a semantic space. Now, you run into all sorts of problems when you do this sort of trickery. One is that, visual space is easy to visualize because vision, right? But semantic space, kind of hard to visualize. So those of you that are really into AI, that there's different types of ways in which you can you can partition semantic spaces, right? You have like word to vec, you have different things. Now, what these authors did is they used WordNet, which some of you may know from AI methodologies, very standard way of it's like a hierarchical way of thinking about concepts. And that hierarchy you can show as a tree. And so that's what you're seeing here. You have like very basic concepts like organism is very central, right? So, and you see that many things stem from organism, like person and like animal. You have like a thing, like artifact, right? It's also very central. It's sort of attached to organism, right? Because, organisms are artifacts, sort of thing. And you see all of the things that we have, like device or tools, they all stem from artifact, right? So it's a hierarchical way of thinking about semantics. Yeah. But that also means that if you have this type of tree-like organization, what you can do is actually plot a receptive field in this structure. So what you see here is actually a receptive field from the parahippocampal place area. So this is a region that we usually map out by showing people images of places and then of faces, and then saying, oh, what regions activate more for places than for faces, and you get this region. And what you see is that in this semantic space, this voxel really likes things and buildings, but also persons just a bit. right? And you really see, so blue is deactivation and red is activation. So you really see that this region of the brain activates for anything, right? And any building and that sort of thing. And not so much for things like animals, and not at all for bodies and I don't know, materials underwater, that sort of thing. Yeah. So this is just a way of seeing what the receptive field tuning of this cortical location is. And so you really get in there, like, okay, now we have a very rich representation, right? We this is very specific and it's very rich, but how do we make sense of this, right? This is one way of doing it, but you know, you have 100,000 locations in the brain that you can click through. You see this thing all the time, yeah, we just get dizzy, right? It doesn't teach you anything how the brain works. So we need to like take an extra step. So in this case, what these authors did was some dimensionality reduction. I'm not going to go into that. We can talk about dimensionality reduction some other time. But what they could do is like color this sort of semantic space according to a couple of different dimensions. So what you do is you look at how the brain represents these things in total, and then you can color, for example, things like animals. They're going to be all very yellow, yeah? And everything that has to do with persons is going to be green. Anything structure, building type is going to be like more purplish, and then things like things that you can travel in. Travel is purple. Boats and cars are also purple, right? So you can sort of color code meaning, like semantic content in this way. Yeah. And then what you can then do is project those components onto voxels again. Yeah? So people are watching a movie, and then what you start seeing is that you actually have these beautiful maps of semantic content in the brain. So what you're looking at here is very similar to that flattened version that I showed you on the beginning of the lecture, right? So visual cortex is here in the middle, and the frontal cortex parts are here at the sides. Yeah. And what you see is that there's, for example, a lot of yellow that happens here, but there are also these parts that are more bluish and reddish there. Which is really cool, because now you have like a sort of continuous semantic representation of what these brain regions like to analyze. Yeah, you get a sort of grouping of okay, what do the semantic receptive fields in each of these regions like best? Yeah. I'm going to quickly shift over this one. It's in the PDF, so you're very welcome to look at it, but I'm going to gloss over it for now. So if you do this type of analysis, you can really start looking at like semantic processing and how it's tiled in the brain, how it's tiling the brain. And for example, what you can then really beautifully see is that certain types of concepts really are processed in a specific location in the brain. And as a whole, of course, then you get the sort of semantic lookup table of what the brain is doing, right? You know exactly what types of mental content are represented in which locations. So you have certain things like mental axis of a semantic space, like how mental is it, or how emotional is it, how social is it. You have other things like how abstract is it tied to location, is it numeric? It turns out, by the way, that if you look at the map for numeric, you actually see these numerosity maps also back. So it sort of all ties together, right? These semantic types of information processing and things that we know from other examples. Yeah.
I was wondering what are the numbers in the areas?
Ah, yes. So in this example, right, which is based on something very, very similar to the movies, they sort of claim that they can create an atlas of these information processing. So they can actually like delineate specific regions where they can say, this is only doing social type processing. And then this one, it's doing like relational processing or that sort of thing. And so these are all like labeled with numbers according to this atlas that they say they've created. Does that make sense?
It's the temporal activation of the different, like there's number 5, for example, activated, for instance,
Oh, no, it's just a label. Yeah, these are arbitrary labels. Yeah. So what you do see here is, for example, 5. If you go to 5, what you see is that it's a bit mental, not so much emotional, very social, then not so much communal, very professional, and very temporal, whatever that may mean. And then it sort of deactivates for things that are abstract and locational or tactile. Yeah? So it's a very warm. So they didn't choose this color map for nothing. It's red in many cases. It's because it's like a warm region. It does social processing. Yeah. Whereas other regions are much more involved in tactile processing. And so this semantic mapping allows us to really say, okay, the receptive fields in these regions, they perform a specific type of cognitive function. Right? So they're using sort of the semantic labels as a way of getting at the type of processing that each of these locations is doing. Yeah? And this is just an example of how you can then start to sort of partition the brain and start to say, well, if you're thinking about your mother, it's probably going to be like a mental, emotional, social thing and not so much a numeric thing, right? You only have one mother or maybe two, or I don't care how many mothers, but you get the point, right? It's a sort of specific type of information processing. So if you have an atlas that generalizes across people, you could even say like, I expect that this pattern of activation on the brain should occur when I instruct people to think about their mother, right? In a very sort of global way. Yeah? But this is just because people like these atlases. I don't so much. It's not very important. I won't. Not an exam question, I can tell you that. But yeah, so this is like just the semantic space that we can use to construct a receptive field idea, right? And this is a way in which we can then start to look at how the brain does this sort of thing.
I've generalizes for all the patients, or it's no patients like all the.
Yeah, so the atlas does, but the specific patterns across voxels not so much. Yeah. So this is the type of thing where you run into how unique people are, because especially at these very high levels of processing, people are quite different in terms of like the functional anatomy of their brains. You can't average them. If you average brains, then
semantics is different. For example, I think about my mother, I might think that is something comfortable and lovely, but if my mother beat me when I was young, maybe it's like violence. So it's a different.
Exactly. Yes. So, and that's just the meaning, how it may be different, like subjectively for people. That even is apart from the fact that your brain organization is probably also much different. And then it's very unclear how your difference in brain organization with, for example, my brain organization relates to that difference between how we see our mothers. This is going in a weird direction though. But yeah. So I just quickly want to go into this last sort of extension, which is if you have an arbitrary space that you can use to build receptive fields in, you can also use models of the brain as in convolutional neural networks, right? And that's the paper that you've just read actually does this sort of thing exactly, right? And that's really the type of framework that I want you to recall that paper, and I want you to see that that article in also, right? We can just start using a convolutional neural network as an encoding model in exactly this way, where we can see like, okay, if something is in the middle layers of this network, and if a voxel or a location in the brain that we record from, if that corresponds to exactly this node in our network, yeah, then that is like a model, like a receptive field model of what that brain location is doing, what the processing in the brain that happens there is. And you definitely see that this architectural thing that we know, like okay, the convolutional neural networks are sort of architected to look like a brain in terms of this hierarchy. You really see that really is the case also, right? So the lowest layers are in the lowest levels of the visual system. So that's all the way in the back of the brain. And then as you go to higher layers, you have higher brain regions, higher visual brain regions that correspond best to these higher layers, right? So this is just two ways of showing you exactly the same thing. Like a V1, activations in V1 are very similar to the lowest layers of these convolutional neural networks. Whereas things like lateral occipital cortex, which is very much related with like object-related processing, like telling a pair of scissors apart from a knife, that sort of thing. You see these regions activate, and that's a type of activity that you would associate much more with the close to the fully connected layers of the convolutional neural network. A much more complicated high-level type of processing. So you see that this analogy pans out. And we can talk in more detail later when we discuss things about podcasts and things like that. All right, so I just want to a few sort of take-home things, just thoughts. What's really cool is, of course, if you have this type of model for a brain activation and you use this sort of receptive field analogy, you can start to really compare different computational models in their efficacy in explaining brains, right? So you can really start to see, okay, which of these models is the more brain-like model, for example. Yeah. And so you've gone through the reading assignment just now. There's a few more novel papers that are coming out. If you're interested in learning more about that, I can send you a couple of papers. I don't want to put them in the course materials. They're not exam materials. But if you're interested in like that paper that you read this week, there are other papers that I can send you for that. You can also start to look at things like attention, right? I might give you an extra lecture on attention at some point, but cognition, right? Complicated cognitive processes, things like learning, value, all that sort of types of processing, you can start to see in this framework and then start to understand as to how it maps onto brain activations. What type of processing really is the type of processing that the brain performs. And then one thing that I really like about this is that fMRI, it's also my field, right? It's sort of a segue, but it used to be this sort of blobology thing. Oh, we have this blob here in the brain and it does this. So yeah, whenever, it's a very vague, sort of weird psychological notion of using brain imaging. But this is really a way of using computational models to start to make sense of fMRI. And then, you're really able to do neural models in humans, which is a really powerful approach. Yeah, that's it for today. We'll see you on Friday for the practical, if that's your kind of thing. If you're already finished with the practical, yay. Please do send me your preferred podcasts by the end of the week so that I can start divvying things up, and that gives you enough time to get together with the group and also start preparing the presentations. Yeah. If you when the practical starts, I won't be there. I'll be late for the practical because of personal stuff. But Ron, for example, and Shifan, who's not here now, will be guiding the practical when you arrive. Yeah. Will it be what? Oh, will sure. Yeah. Ron, can you record it? He can.
I think so.
Yeah, that should be good. Yeah. Ooh, there's a chat that I missed. Oh yeah, yeah. So that's indeed Ben Harvey. Yes, I saw a presentation at the conference I was at by the person you're referring to, and yes. It is working memory load seems to be the same. Sorry, just yeah. Good comment.
And also show that it's numerosity like captured by Fourier thing, right? This aggregate Fourier.
Yeah, yeah. There's specific things in the visual stimulus that are numerosity. Yeah. That relate to numerosity.
That was when it goes from 1 to 20, so